Wow, that was pretty amazing. Everybody appearing all at once. Hi, John. Hi, everybody.
Hi, guys.
Let's get started. Welcome to the ARO seminar series today entitled Illuminating Inner Ear Development, Function and Protection with a Multi-Omic Torch. My name is Stefan Heller, and I will be your moderator for today's webinar. I'm a professor at Stanford School of Medicine in the Department of Otolaryngology. I'm also affiliated with the Institute of Stem Cell Biology and Regenerative Medicine at Stanford. My laboratory is interested in regenerating hair cells, and we are innovative in our research, trying to use the latest technologies to leapfrog ahead whenever possible. I'm very excited to host today's webinar for you because Dr. Ronna Hertzano, whom I have known for many years now, has been equally excited about implementing new technologies, particularly omic-based approaches. Ronna has made substantial contributions to our field, and her lab is at the forefront of pushing advances in inner ear biology with, in my opinion, unprecedented speed. Dr. Hertzano is a professor of otolaryngology, head and neck surgery, University of Maryland School of Medicine. Her clinical practice focuses on the diagnosis and treatment of diseases of the ear with an emphasis on hearing restoration. Translationally, her goal is to make significant contributions toward the treatment of congenital and acquired auditory and vestibular dysfunction. Toward hearing restoration, she works to unravel the regulatory signaling cascades that lead to proper development of the ear and specifically the hair cells. She leads a collaborative team that develops and applies a variety of approaches for cell type specific multi-omic analysis of the ear, followed by state-of-the-art informatic analysis to identify key regulators of gene expression in hair cell development and cell type specific signaling cascades in acquired hearing loss, such as noise-induced hearing loss. To facilitate dissemination, sharing, and analysis of multi-omic data, Dr. Hertzano is the founder of the GEAR portal, Gene Expression Analysis Resource, at umgear.org. Finally, Dr. Hertzano has a strong interest in mentorship and training of the next generation of clinicians, scientists, and clinician scientists. Before we get started, I would like to review a few meeting tools that you as an attendee will be using throughout the presentation. These tools are located at the bottom of your screen. To keep questions from becoming distracting to our presenter, please utilize the chat function directly at myself as a moderator, or if you post it to everybody, I will keep track of it. You're welcome to submit your questions at any time throughout the presentation by typing into the chat function and clicking the send button. Any questions that are not addressed during the session will be answered by the presenter in a follow-up email. Closed captioning for this meeting is available for your use, and the session is being recorded and will be made available for additional viewing later. We hope you will also participate in the post-webinar evaluation so we can gather feedback. Also, to be sure to join us on Thursday, December 15, from noon to 1 p.m. Eastern Time to meet with the Executive Office staff and ask questions about the 2023 midwinter meeting. Lastly, if you have technical difficulty, please email headquarters@aro.org for assistance. And now, without further delay, I would like to introduce Ronna. Here you go.
Yeah. Thank you, Stefan, for the extremely kind introduction. Thank you all for joining us here for lunch. I appreciate you taking the time. And I will start. As mentioned, the topic of the talk today is going to be illuminating inner ear development, function, and protection with a multi-omic torch. And this is, as Stefan mentioned, this is a shared passion of not only him and I, but many other investigators in our field. I have no significant conflicts to disclose. So I think it all starts with hearing, right? When we're born, the range, our sensitivity for hearing far exceeds what we need for communication. This is an audiogram, and you can see that the thresholds that we have up to age 20, give or take, far exceed where the consonants or syllables are pronounced. And really, we use only up to about 4,000 hertz for speech. But with normal healthy aging, even at a young age as 56 to 65, our thresholds change dramatically. So having hearing loss is not something that is unique to people that have specific mutations, but it is normal to have hearing loss. Only that with normal healthy aging, our thresholds start to increase to a point that they interrupt with speech understanding. And if we were able to image an ear of an aging individual, much of the damage would probably be in the outer hair cells. So it gets to a point that by age 70, 1 in every 2 individuals should be using a hearing aid. And according to the World Health Organization, there are 430 million people worldwide with disabling hearing loss, and it is estimated that by 2050, over 700 million people will have disabling hearing loss. Now, hearing loss is so common that if you went to this website, how old are your ears, you would be able to determine your age by just different sounds that are played. And you can see that there are characteristic frequencies that we lose our ability to hear as we age. But what's really interesting to me is that no matter if our hearing loss is from normal aging, from noise exposure, from genetic mutations, chemotherapy, infection, or other drugs, we only treat it based on how bad it is. If we have a little hearing loss, we treat it with hearing aids. And we have a little bit more hearing loss, we treat it with cochlear implants. And as a clinician scientist, I'm very passionate on trying to figure out, can we reverse hearing loss through, for example, hair cell regeneration? Or even better, can we stop this process altogether? Now, omic analyses allow us to take an unbiased approach for discovery, because when we take an approach through omics, basically we ask the tissue to provide us with the answers. And when we think about hair cell development, we know about 3 key transcription factors that are important for hair cells formation. And when we put these 3 transcription factors in supporting cells during normal development, we will get hair cells. However, when we take these 3 transcription factors, ATO1, PAL4, and GFI1, and put them in stem cells, we get only hair cell-like cells, which look quite different from the mature hair cells. And one of the questions that we ask then is, how can we advance this process? So to advance this process, we need to identify additional regulators of inner ear cell type specific differentiation. And we did not want to just look at lists of genes, identify transcription factors, and go one transcription factor at a time to try to understand which one has an important role. Talking with my long-term collaborator, Ronna, we've been working together now for 2 decades. We surmised that if you take any 3 cells, or the same cell at 3 different time points, or before and after noise exposure, or any other combination of conditions, there are going to be many genes that are going to be shared in their expression. So if you think about this red cell as a hair cell, the yellow cell as a neuron, and the blue cell as a blood cell, only the neuron is going to have axon and dendrites. Only the hair cell is going to have stereocilia. And those genes are going that form the unique structures are going to be unique. If we would look at the upstream sequences from each one of these genes that are unique to the cell type compared to the genes that are shared in the cell type with other cell types that perhaps are expressed in the same tissue, we should be able to find short sequences that are enriched in the regulatory regions and the upstream regions of the genes that are uniquely expressed in a cell type or in a condition. And that is because programs tend to be regulated by common transcription factors. And these short sequences would be the parking lots for the transcription factors, or what is called the binding sites. So with this in mind, if we are trying to understand how can we promote a hair cell-like cell to become a more mature hair cell, perhaps we should look at the gene expression in hair cells at early postnatal stages and try to see if the hair cells in early postnatal stages have any short sequences that are uniquely overexpressed in the hair cell specific genes compared to other genes that are expressed in hair cells but are not unique to hair cells. Well, that is a neat idea, except that when we look at a cross-section of a newborn cochlea, you can see here that there are a lot of different types of cells. And the hair cells, you can see here 3 outer hair cells and 1 inner hair cell, are less than 1% of the cells in this cross-section. If we wanted to learn about the gene expression just in hair cells, we would need to come up with ways to isolate specifically the hair cells. Now, nowadays, we can use single-cell RNA-seq, and we'll get to that. But when we started these projects, single-cell RNA-seq was not available. However, we were aware that there are cell surface proteins that are commonly used in immunology called CD genes, for which there are antibodies. And when we looked at a microarray that we did, we found that over 100 of them were expressed in the ear. And when I started putting antibodies for these different CD genes on sections of newborn inner ears, I was able to see that different CD genes had different patterns of expression in the ear. And as an aside, in the ARO that I presented this work for the first time, Stefan Heller also presented an almost identical project in the same session. So we both had the idea and came to the same conclusion at the same time. And so we were both able to take these CD genes and put together slightly different puzzles, but very similar, where we CD326 that targets a molecule called epicam can be used to separate the ear into epithelial and non-epithelial cells. And with the use of other CD antibodies, we would theoretically be able to isolate neurons, blood vessels, and mesenchyme. And when we took these antibodies at the common concentrations, dissociated the ears, and then put them through the flow cytometer, we were able to see that indeed the inner ear was divided into 2 big populations that should be epithelial and non-epithelial, and with the additional antibodies added, into the predicted distribution of cells. To find out if these indeed were the cells that we were predicting, we marked circles around each population and collected them into tubes, extracted the RNA from the cells that we collected, and did a microarray analysis. And this analysis identified groups of genes that were unique to each one of the predicted populations, and based on their gene names, we were able to confirm that indeed we had sensory cells, neurons, blood cells, and mesenchyme in the cochlea and the vestibule. And as a test, we then went ahead and we took the genes that are expressed uniquely in sensory epithelium, in the cochlea, and in the vestibular system. And we asked the question that we told you about before. We asked, is there any sequence that will be enriched in the promoter regions of these sensory epithelium enriched genes? And when we did this analysis, it came back with this short sequence. And this short sequence was a perfect match to the binding site of a transcription factor named ZEB1. ZEB1 is an inhibitor of expression, so it should be expressed in all cells that are not epithelial cells. And you can see here that it is indeed expressed in all of the cells in the cochlea, marked in red, that are not epithelial cells. This was very interesting, and it was actually in an ARO that I met Andy Griffith and told him about this work, and he mentioned that he actually has a mouse with a mutation in ZEB1. So we obtained the mouse, and we found that together with him that the mouse had severe epithelial defects, and in fact, in cross sections, we were able to show significant alterations in gene expression in epithelial genes. So that showed us that this approach can work, and we wanted to proceed and focus on hair cells. The problem with hair cells was that there was no good antibody for hair cells that would target the extracellular domain and would work well for us for cell sorting. So we combined our Otosort approach, which is CD326, with a mouse that expresses a green fluorescent protein in all hair cells, the ATA1 and GFP. And when we did that and took the dissociated cells to the flow cytometer, we identified three main populations. There was a population that was both epithelial and green. This should be the hair cells. There was a population that was epithelial but not green. This should be epithelial cells that are not hair cells. And there was a population that was all the rest. And we took a small sample from each population, ran it again, and saw that we're getting pretty good purity. Went ahead, collected the cells, extracted RNA, and continued with analyses. When we looked again to see is there a short sequence upstream of the genes that are specifically expressed in hair cells, we found an enrichment for a motif that we've previously not heard about in the context of the ear. It was the binding site of a family of transcription factors named RFX. This family of transcription factors would have been quite a bit of work to study because they had more than 8 transcription factors, and we might need to knock out several of them to get an effect. To convince ourselves that this is indeed important, we went ahead and we took the regulatory sequences of the orthologs of the hair cell specific genes from human and from zebrafish, and we repeated the analysis. And when we saw that when we repeated the analysis, again, the same regulatory sequence came enriched, we were now convinced that RFX may have an important role in hair cell development. We went ahead and we generated either conditional knockouts or knockouts for RFX1, RFX2, RFX3, and RFX7. And as you see here, in red are the hearing thresholds of the mutants, and in black are the hearing thresholds of the wild type. None of these models had an auditory phenotype. RFX3 and 7 have the highest expression in the ear, and when we made a double knockout for them, these two did not have a phenotype. But RFX1 and 3 are in the same subgroup. The RFX family of transcription factors are divided into groups of transcription factors, and 1 and 3 have been shown to work together in the pancreas. And when we deleted these two, at 3 months, the mice were completely deaf. We went ahead and we took the ears of these mice and performed scanning electron microscopy. And we found that at 3 months, the RFX knockouts had no outer hair cells, and the inner hair cells had significant alterations, and many of them were missing. This was very exciting, but it suggested that we missed the critical point. And indeed, the analysis that we did to identify the RFX transcription factors was done at P1. And we're looking here at 3 months. So we started looking at earlier time points. So first we look when the mouse was 3 days old, and we used the FM143 vital dye. And we saw that there was no difference between the wild type and the mutants. The dye was internalized, suggesting that the transduction channel was present in the absence of RFX1 and RFX3. Then we looked at Preston expression. Preston is a marker of the outer hair cells and is consistent with outer hair cell differentiation. It is important for the electromotility of the outer hair cells, and it was present too. Then we did scanning electron microscopy And we weren't sure that our scanning electron microscopy was sensitive enough, so we asked Gregory Furlenkov to repeat and do scanning electron microscopy. Maybe we're missing something, but he too could not identify any changes. So we went on and we looked now at P12. P12 is a day before the onset of hearing in mice. And here again, things looked largely normal, except that we had occasional missing outer hair cells. We looked then at P15, and the difference between P12 and P15 was striking. The outer hair cells were almost completely lost. There was an increase in apoptotic genes. And this meant that the RFX transcription factors are very important. But now we did not know, are they regulators of terminal differentiation? And because the hair cells are missing something, now they can't withstand sound. Or are they regulators of hair cell survival? And maybe more RFX will be helpful to protect, for example, from noise trauma. So we went ahead, and this is all unpublished data, and did a series of analyses, now taking different Cree drivers and deleting the RFX transcription factors at different time points. We already knew that losing RFX 1 and 3 at E16 will cause hair cell death at P15. We next deleted RFX 1 and 3 at P21 using the Preston Cree RT2. And to our surprise, nothing happened. Hearing thresholds were normal even at 3 months of age. We went ahead and deleted them at P9. And again, nothing happened. When we deleted them at P0 with the myosin 15 Cree, we had a similar phenotype. So this means that the critical window for RFX 1-3 for hair cell differentiation is at terminal differentiation between E16 and P9. And in the ARO, two of our students, Katie William and Ninjin Wu, will have presentations about the RFX transcription factor. Katie about the role of RFX2 and the role of these transcription factors in the vestibular system, and Ninjin about RFX 1 and 3 in the cochlea. Now, part of the work of Katie uses again omics as a genome, as a flashlight to figure out what is going on. And we did not want to make any early assumptions onto what causes the vestibular phenotype. And we wanted to identify the downstream signaling pathway. So Katie harvested vestibular organs, added the CD326 epithelial marker, but then rather than sorting the hair cells, say with FM143, realized that the hair cells might have many different phenotypes. We have type 1 and type 2 hair cells. The hair cells in the vestibular system, some of them are strialar, some of them are extrastrialar, and all of these might have very different phenotypes. So for this reason, she collected the CD326 positive cells, processed them with 10x genomics, and performed the data analysis herself. And to find more about this, please come and visit her poster at the ARO. So moving on to a different story, we were also interested in understanding, can omics identify regulators of outer hair cell terminal differentiation? And outer hair cells are not easy to study, particularly in the mature phase, because when you think about dissociation is a significant trauma, and outer hair cells are very sensitive. And in our hands, at least, when we dissociate mature ears, we cannot get very large number of cells. So this is a project that was led by Maggie Matern from my lab that is now a postdoctoral fellow with Stefan Heller, where we used the ribotag model that when it's crossed with a cell type specific Cree, will result in the ribosomes of that cell having a tag on them, specifically an HA tag. And that means that if you want to get enrichment of transcripts from a certain cell type, the cell type that expresses the Cree recombinase, all you need to do is to then freeze the tissue, and you can immunoprecipitate the RNA that is bound to the ribosomes. When Maggie did that at several different time points in development and in mature ears, she found an enrichment of 5 transcription factors, one of which was uniquely expressed in outer hair cells. These images were generated actually by Lauren Chesham from Mike Bowles' lab. Mike is a collaborator and a friend that was at the time at the MRC Harwell. And he, at the same time, found a mouse model that had hearing loss through an aging screen. And this mouse was called Cello. It had a mutation in the IKZF2 transcription factor. It had elevated hearing thresholds. It had absent DPOE. DPOE are distortion product otoacoustic emissions that are generated by the outer hair cells. And the mouse had the outer hair cells, but the outer hair cells had very low electromotility. This was all consistent with outer hair cells requiring IKZF2 to possibly terminally differentiate. To figure out if this is indeed something that happens through a transcriptomic pathway and gene regulation of the IKZF2, we took the IKZF2, put it in an AAV vector, and injected it into a mouse that expresses a red fluorescent protein in the hair cells. We then collected these hair cells and, together with the lab of Matt Kelly from the NIDCD, performed single-cell RNA sequencing and analysis, first identifying markers that are canonical for inner and outer hair cells, both in control injected and in IKZF2 injected. But then we wanted to look and see what happened to inner hair cells that now got what we think is a regulator of outer hair cell fate. And when we did that, on the left, we always have here the controls, and on the right, we have the IKZF2 injected. You will see that genes that are normally highly expressed in inner hair cells, when they had IKZF2 in them, they were significantly decreased in expression. And genes that are normally uniquely expressed in outer hair cells were now all of a sudden higher in their expression in the inner hair cells. So IKZF2 in the inner hair cells was turning down some of the inner hair cell characteristic genes and turning on some of the outer hair cell characteristic genes. And then there were some genes that it was not changing. And this did not happen only at the messenger RNA level, but when you look at the protein level, this is a control uninjected ear 2 months after injection. And in the injected mice, you see that there was expression of Prestin throughout the length of the cochlear duct. So the big question is then, what is exactly IKZF2 regulating and how we're going to find that? And this was a little challenging because we did not want to now go and cross this mouse with yet another mouse with a fluorescent marker. And we had to come up with a method to allow us to isolate outer hair cells specifically without yet another transgenic model. So this is work that Beatrice Millen from my lab did. And what she did and suggested is that we'll take the ears and we'll dissect them. But then we will put them in cold HBSS and add the FM143 vital dye. After we do that, that will allow us to potentially see the inner and outer hair cells. But because CD326 has 2 different intensities, in epithelial, non-epithelial, and also FM143 is a little different, we might be able to use the combination of the differences in expression of CD326 and also FM143 to isolate inner from outer hair cells. And we noticed that there was a larger population that were CD326 low and a smaller population that was CD326 high that maybe could be outer hair cells and inner hair cells, and this way really enrich for outer hair cells without using yet another transgenic model. Because if we wanted to really find out what is regulated by IKZF2, and we would be taking all of the cochlear cells or all of the epithelial cells, we would be limited in the capture. And we would not be sequencing enough outer hair cells to be able to make and generate very significant data. So this was our assumption, and Beatrice circled the cell population and then took it for single cell analysis. And what she found was that she collected roughly 900 outer hair cells, and about 95% of them expressed the OCM marker, which is an outer hair cell marker. And just a very small number of them expressed an inner hair cell marker, showing that this is really a very good method to enrich for outer hair cells without another transgenic line. So, of course, we implemented it to identify the downstream signaling pathway of IKZF2 using both bulk and single cell RNA-seq. And if you want to find out more about it, please come to Chris's poster at the ARO, the Helios transcription factors necessary for outer hair cell development and maintenance. But hair cells are not the only important thing in the cochlea. If we take a step back from the hair cells, the otic mesenchyme is actually the most common cell type in the developing inner ear, and it's also the least studied. Nevertheless, there are several transcription factors in other genes that are expressed in otic mesenchyme cells, and when we have mutations in them, humans suffer from hearing loss. So both Tom Coate from Georgetown University and I were very interested in PAL304 and decided to collaborate on this project. And the person that did most of the work is Kevin Rose from my lab. And we wanted to find out, what are these mesenchyme cells, what regulates them, and what does actually PAL304 do in the mesenchyme cells, except that we didn't even know if mesenchyme cells are one or more populations. So to do this, again, coming at a very unbiased approach, the first step was to do single cell RNA-seq. And Kevin was able to show, using single cell RNA sequencing, that the mesenchyme cells are actually 4 different populations. And these 4 populations, not only are they completely distinct with very unique markers, but they're also spatially distinct. And they're expressed in different areas. So type 4 is along the lateral wall, type 2 is in the spiral limbus, type 1 is the basilar membrane. And when he also looked at the PAL304 mutant mice, he was able to show that PAL304 regulates a completely different transcriptional cascade in each one of these mesenchyme subpopulations. So to learn more about the mesenchyme cells and what we learned using omics, please come to the poster of Kevin and Gabby from my lab in the ARO. And a lot of the methods that we used, we summarized recently in a technical report. But until now, we spoke about inner ear development. Can we apply cell type specific omics to identify new candidates for therapeutics? Functional studies provide mechanistic cues to pathways that underlie noise-induced hearing loss and age-related hearing loss. We know that noise exposure results in outer hair cell dysfunction or outer hair cell loss. We know that noise exposure results in swelling of the lateral wall and lowering of the endocochlear potential. We know that there is axonal retraction of the auditory nerve fibers, resulting in loss of innervation of inner hair cells. And we also know that there is activation of the immune response. This means, yet again, that each cochlear functional domain has a different response to noise and should be studied separately if we really want to understand noise-induced hearing loss. And this seemed like a very big task. And we decided to set out to construct a transcriptional map of the cell type specific response to permanent threshold shift inducing noise to better understand noise-induced hearing loss. And to do that, we worked with a lot of friends from academia and also from industry. And what we did was we took adult mice with normal hearing in each one of the different locations. We exposed the mice to a noise exposure that will cause a permanent hearing loss across all frequencies. And then in each location, we used a different cell type specific omic approach, whether it was ribotag, single cell RNA sequencing, or single cell RNA sequencing with flow cytometry, to understand what are the gene expression changes in each one of the cellular population in the ear. And we did a combined analysis. And at the end of the analysis was published recently in Cell Reports. You can learn more about the different changes in gene expression in each one of these populations there. The data are also available in GEAR. We wanted to identify candidates for drug repurposing to prevent noise-induced hearing loss. So we combined our data with Drug Central, and we looked at drugs that will have an opposing effect to the effect on gene expression as a result from noise. And here are our top candidates, where we found it very interesting that halogenated compounds that are routinely used for anesthesia were actually predicted to be very protective. And this is really interesting also from a translational standpoint, right? Because, for example, if we're doing a cochlear implant for hearing preservation, there's no normalized anesthesia protocol. But it is possible that if we chose specific anesthetics, we could potentially have a better protective effect on the hearing. However, the top compound was metformin. And metformin was introduced as glucophage in 1957. It's approved by the FDA in 1994 to treat diabetes. It does not have an effect of hypoglycemia if it's used to treat people that do not have diabetes, and it's associated with lower rates of sudden hearing loss in patients with diabetes. So to learn more about whether metformin that we identified as a candidate drug can prevent noise-induced hearing loss or age-induced hearing loss, you can guess the next slide. Come to our poster at ARO, where we present our work. So the conclusion of this work was that the inner ear response to noise is cell type specific. We have some immune molecules. We have very interesting pathways that are good for pursuing in the different cell types. And we also have several drugs for repurposing. And this all has been published recently. So until now, I basically showed how we use omics as a flashlight for discovery, but not only us use it, many people in the field use it. I think at this point, almost all molecular biologists in the field use it. And there are many questions. For example, how can you get access to published data? How can you understand and visualize single cell data if you're a biologist and you're not a bioinformatician? How can you view your data privately in the context of published data? And how can you make cross-dataset comparisons? And for that, back in 2014, we started working on the GEAR, which was very, very generously funded by the Hearing Restoration Project of the HHF, and more recently picked up by the NIDCD as an R01. So this is the URL for the GEAR, for those of you who have not seen it and are key players with Joshua Orvis as our lead engineer. It's a platform that is continuously being developed. You can put in any gene name or groups of gene names, and it shows you the expression and groups of data sets. Every box is a data set. You can learn about the genes. You can go to outside websites. You can learn about whether a gene causes hearing loss in a mouse model or human and go to the models. There are different tools, including for analysis of the data. You can perform single cell analyses without coding. And the GEAR now actually satisfies yet another important need. Starting from January 25, 2023, according to scientific data sharing mandate, it is necessary to manage, budget, and plan for data sharing in all grant applications. So for the ear field, this is not only a one-stop shop for depositing the data, but also for making it very accessible for users. And I would like to put here a plug for the gear workshops. There'll be many of these in the ARO, and the gear is going through a major shift and uplift before the meeting. So please join us for the gear workshops in this coming ARO. So with this, I'd like to leave a few minutes for questions, not before thanking the members of my team, all of which are in this photograph here. Beatrice Millen is our research supervisor who helps me oversee all of the projects. My collaborators at University of Maryland, Mike Bull, now in UCL, many collaborators I've mentioned throughout the talk are very generous funding. We have an opening for a postdoctoral fellow, and I would like to thank all of you for taking time from your day to come to this talk. I'm happy to take questions.
Thank you, Ronna. That last slide, that was a fascinating talk, first of all. It was really encouraging to people, I think, at least to me. That last slide showed that you really engaged in so many collaborations and that they are really fruitful because you publish with everybody on that slide. People sometimes show collaborators that are only there pro forma, but you really do this. So here our first question is there a plan to add data to gear from species other than mouse and human? There is data.
Yeah, so we actually have data in gear right now from chick and from zebrafish in the cochlea. A lot of the chick data came from Stefan's lab, zebrafish from David Rabel's lab, from Tatiana Petrovsky's lab, and other labs. The gear does support also non-human primate data, and it supports rat. And if there are, essentially, if there is a data set on a species that you've generated and we don't have support for it, send us a note and we will make that supported.
Cool. I have a question regarding the data you showed with RFX 1 and 3, which is really intriguing. So if you knock out these transcription factors after postnatal day 8 or 9, everything is normal. So that tells me that once the hair cell is built and whatever these transcription factors are accessing in the whole building block repertoire of building blocks that are needed, then later the hair cell doesn't need to add these as spare parts anymore. So that leads me to the question of turnover. Is this going to be an indication that an adult hair cell turns over very little?
I don't know. That's a great question. I think one of the questions is, great, what is turned over? Because there may be that there is turnover in the in areas that the RFX transcription factors are not regulating. Or the other option is that there are things that are necessary early on for the hair cell to turn on certain projects. Maybe RFX is just regulating yet another regulator that turns on another process or project, right? We were very much expecting the RFX transcription factors knockouts to have a planar cell polarity defect because they regulate celiogenesis in other systems. And we looked very carefully and we did not find that. So hopefully the data Katie will show, and she's still in the process of analyzing them, will identify what they regulate, and we'll be able to answer your question.
Mm-hmm. Will be cool going to her poster presentation at ARO. There's another question that is any functionality change in inner hair cells after IKZF2 was transduced in inner hair cells? Do they change their function?
They acquired electromotility.
Very cool. Waiting for other questions. What kind of tips would you give a regular lab that produces data? How would they deposit the data in gear?
That's a great question. So there are several levels of depositing data in gear, and my first thing would be if you're a lab that does omics or uses omics as a technique, the gear is not there just to share data post-publication or for publication, but the gear is there to help you even the moment you got the data off the sequencer. When you upload data to gear, you can choose if your data is private or if your data is public. And if you upload the data and you upload it private, then you can use all the tools the gear has to look at your data and to study it. You can have your data and another 2, 3, 4, whatever number data sets that are from the field side by side on it in the page. And if our tutorials are not clear on how to do that and how to upload, we're actually funded to help you upload your data privately. We can also generate private spaces in gear for teams. So for example, there are several teams in the field that whenever they log on, anybody in the team has access to yet a whole new menu of data sets that were generated by their team. Nobody else has access to those, but then you can look at data sets that were generated by your colleagues in the lab, or you can share it with your collaborators that way. And again, we're funded to support you to make these spaces. The next step of uploading here, and you can upload many times, and as your analysis continues, you can upload more advanced versions of it. But the next step is you're ready to submit your paper for publication. You can generate short links that you can use for the method section, or you can use for the figure legends, so that the reviewers can then click on the link and they can actually interact with your data. And ultimately, once the data is published, the readers will be able to not look just at the 1 or 2 genes that are shown in a figure, but they will be able to use the gear to interact with your data.
That's pretty cool. Coming back to the inner hair cells, there was a question about, besides becoming electromodile, are there any studies you have done with respect to changed innervation patterns of these cells?
We have not, but these are really interesting studies to do. We also think that it's worth looking more carefully at the knockout outer hair cells for innervation. So that is a very good question.
It seems that your talk was ultra clear, and so we don't have a lot of questions. I think there are lots of experts in the audience, which is great. And as a Japanese saying is, sometimes you can stop eating before you're completely full. So we all get back 10 minutes of our time of the day. And if you have additional questions or something pops in your mind, I'm pretty sure Ronna will answer all these questions, and email her directly and find her at ARO. She's always good for a good chat. With that, thank you again. I got comments that this is an amazing talk. Congratulations, and see you all. Have a good holiday season, and hopefully see you all at ARO.
See you all, and Stefan, thank you again.